We are going to get started. Thank you so much for being here today at today's seminar. My name is Cinnamon Moffett, and I'm the Research Program Manager here at Oregon State University's Hatfield Marine Science Center. This is a hybrid event, so we have folks that are online and folks that are in the room. Thank you all for being here. For those folks that are online, Roseanne is your hybrid support tech person today. So if you have any technical issues, make sure you put them in the chat, and she'll make sure that she lets me know if we could do something to make your experience better. For those folks in the room, we will be doing questions at the end of today's talk. Because it's a hybrid event, you will need a mic to ask the questions. So when our speaker today gets to the end, if you have a question, just raise your hand. I'll bring you the mic, or you can come to the mic stand there, and then we can ask those questions. For folks that are online, you have two options. You either can type your question into the chat box, or you can raise your digital hand and Roseanne will unmute you and you can ask your question verbally, and we can hear that happening here. So wanted to also make a couple of little announcements. We are recording today's event, so it'll go on to our past seminar page in a few days. So if you missed something or you want to share it, feel free to do that. And I wanted to make an announcement for next week's seminar. We have Hannah Glover, who is going to be talking to us about managing mud and mangroves. And she is with the civil and construction engineer at Oregon State University. So we're excited to have that perspective coming our way. But for today, I don't think this individual needs any introduction. I think everybody here knows our speaker. But I will just do my little part and introduce John Chapman. John Chapman is courtesy faculty with the Department of Fisheries and Wildlife and Conservation Sciences at Oregon State University, working primarily here at Hatfield. John focuses mainly on aquatic biological invasions and has done a massive amount of work tracking the population of Eupogibia, our local mud shrimp here. And he's going to be talking mostly today about a story about a recently retired research vessel. So I'm going to hand it off to John. Yeah, make your mic go live.
Oh, thank you. Thank you for being here. Thank you for listening. This is I've been gone for most of the last 2 years, and I come back and I subscribe to the local newspaper. And I don't know, sometime back in November of last year, I just saw this headline: "The Oceanus retired." And ships are these iconic things that people. Here's a Marine Science Center. Here's what is a Marine Science Center? It's a place where there's seawater. Hopefully there's a library, there's faculty, but there has to be a boat. And many boats. And so an incredibly iconic thing about any big marine lab is to have a ship that they can do oceanographic research and all kinds of research. But the Oceanus is sort of this iconic thing that has represented the Hatfield Marine Science Center, Oregon State University for the last decade. But it replaced, before that, the Yaquina, that was the sister ship to that was also built in 1975. And that the Yaquina was retired in November of 2011. And that the replacement was to be the Oceanus until we get the new ship that NSF is building for to come here for a much longer term. And so, but the Oceanus was in better shape than the Yaquina. And so the result of that was that it was sailed around from Woods Hole, where it was stationed, to Newport, Oregon. So I've worked for a very long time with a very dear friend who's probably watching this remotely, Ralph Breitenstein. And Ralph had this, and he always is incredibly good at coming up with good ideas. And so he thought it would be a good idea to look at the Oceanus, the bottom of the Oceanus that was going to sail straight from Woods Hole to Newport, Oregon. So I'm an invasion ecologist, and I study introduced species and how they get moved around the world by human activities. And before there were ships, the world was divided up into these, and it still is to somewhat, these geographical boundaries where you would know if you're in Australia or in the Orient or in the Palearctic or in Ethiopia or South America or Australia or in North America just by going outside and looking around. And so these are called Wallace's lines, and they were decided upon or realized in the 19th century. And so, and what ships have done and people and their activities have connected the world. And so if we look at the shipping patterns around the world, every port is connected to every other port on the planet. In fact, this is just the shipping that in fact, this is more than reuniting Pangea. This is every single place where humans occur, something else can occur there. And so this is a very significant thing for Homo sapiens. We've spread our ecosystem and the things that are associated with this everywhere we go. But the study of biological invasions, how things get moved around the world, didn't just happen as soon as we started doing that. Zoology wasn't invented when we started moving things. And so this rather long quote from Chuck Darwin in 1854, he studied barnacles to gain credibility as a biologist. And so even to this day, his monographs are on barnacles, are very important documents for people who study marine biology. And one of the things that he wrote is this section about it should be not overlooked that these species as Balanus, Tujumatsulum, Amphitrite, improvisus, and to a lesser degree, B. trigonus, and I've highlighted that in yellow because I'm going to talk about that, and Tetraclida radiata, which seem to range over nearly the whole world, excepting the colder seas, are species which are habitually attached to ships and which could hardly fail to be widely transported. Indeed, it appears to me surprising that such species as Balanus cetaceus and eberneus, which often become attached to vessels, should still be confined to the one southern and the other, the northern America. So that was of interest, more than 100 years ago. And then in 1988, Jim Carlton, who's a co-author on the paper that I'm going to talk about now, did a study where a replica of the Golden Hind, Sir Francis Drake's vessel, sailed down the coast, and he had fouling plates and scrapings from the Golden Hind, and he tracked it into 4 major ports. It was in the Columbia River in freshwater where everything got killed off in the bottom, presumably. Then it went to Tillamook Bay, and then we received it here in Yaquina Bay, and it was here for about a month. It was in Tillamook Bay for several weeks. Then it was in Coos Bay for about a month. Then it sailed to Humboldt Bay for about a month, and then it went to San Francisco Bay. And in every single place that the Golden Hind went, and it only sailed at 4 knots. So this is like 17th century, 16th century transport of organisms. And it was very impressive how many things he found on there, and that these species that were picked up in one port moved to another. But this is, I don't know, but is probably not the right word. This is one coast. So we might think that the species that are transported along this coast are not as consequential as species that are transported from far away. But it was very impressive that the survival of the species between the ports and the sheer diversity of species that they could find on something that was a replicate of what was going on before there was zoology, before people were actually naming things. Okay, so the story here begins that I'm going to talk about 4 barnacles, but I'm really going to talk about 2. But this is from a publication in 2011 by Jim Carlton. And it's just the introductions of barnacles from the Pacific to the Atlantic shores of the New World. And I'm underlining in red species that was above that Darwin mentions, eberneus, right? So it's there, and he's saying, how could it be separated in these places? Well, it's already here now. So it has spread since he looked at it. And so, and now in the blue are species that are of question. So Balanus Amphitrite is something that's from the Indo-Pacific, and I'm going to talk about it very little, but Balanus trigonus, the triangle barnacle, is something that was in this coast in the Pacific Ocean, and that was introduced into the Atlantic Ocean by the time Darwin was studying barnacles. And he recognized that, he saw that. And so this process of moving things around the world has been there for an extremely long time. As long as people have been moving, pretty much everywhere they go, they take something with them. Okay, so the Oceanus left Woods Hole and sailed around through the Panama Canal and then came to here in about 28 days. Ralph organized, collaborated with these divers from the Oregon Coast Aquarium and from different places and from the Marine Science Center. And here's a very long list of people that came 2 days after the Oceanus arrived to scrape the bottom. It's a very well-maintained ship, very clean, almost nothing there. And these people were extremely thorough in how they did that. And they photographed it. They looked at every single surface. They scraped off everything that they could. And it was an army that went there and scraped the bottom. And in the end, we pulled off the animals, put them in plastic bags, brought them back into the laboratory, sorted them out, and identified them. And what was a very happy circumstance was that Jim Carlton, who was becoming a barnacle expert then, and I'm going to explain that too, was going to be here. And so Ralph and I are good friends with Jim. And so he came and said, I'll identify the barnacles for you. And we pulled them out. And then, and that was the main thing that they found on the bottom of the boat. They found other things, but the barnacles were the predominant thing. But all the barnacles that they could scrape off the bottom of this 177-foot ship wouldn't have filled a bucket. Okay, so what we did was we tracked from the log of the Oceanus that's published online where it had been before it got here. So it was on dry dock in Jacksonville, Florida, and the degrees latitude is 30 degrees, so way south of here. And it arrived there on February 1st, 2009. Then it went back to Woods Hole. Then it went to Piraeus, Greece, and it arrived there in July 26 in 2009, and so on. So then it was back in Hui, which is about 40 degrees, well, it's 40 degrees north latitude, so it's right there, 41. So it would go, the ship, just like here, would sail out of the port and then come back and would go to some exotic port and then come back to Woods Hole Oceanographic Institute. So these places sometimes were at a higher latitude, but almost always it was sailing to a further northern latitude. So these notes over here don't really matter, except that we're just saying, when we inspected it, these are our notes where we thought we saw Balanus trigonus on the ship, the triangular barnacle. Okay, so here's the destinations of the Oceanus before and until it came to Newport. So it went to Galveston, Texas, to Gulfport, Mississippi, to Panama City, Florida, St. Petersburg, Florida, to the St. George's Island of Bermuda, San Juan, Puerto Rico, Bridgetown, Barbados, and it also went to Piraeus, Greece. And so the first place that it left from Woods Hole for an exotic place after it came back from dry dock was in Panama City, Jacksonville. It's not on this map, Florida, back to Woods Hole, then off to Piraeus, Greece. Okay, we'll show you that later. So this is one of the barnacles. This is two barnacles. This is Balanus trigonus, and this is Amphibalanus venustus. And so these were the predominant barnacles that we found on the bottom of the Oceanus. And Balanus trigonus is a tropical barnacle that Darwin noted that's basically all over the tropics in the world. Everywhere in the world, pretty much, this barnacle occurs. Balanus venustus, though, is a northwest Atlantic species of barnacle that's not in the North Pacific. And so, and it lives in nearshore environments, and it settles regularly, and it occurs naturally in the Woods Hole area. So so that's an important point. But the second point is that what was very important about what we were doing is that these settle in layers. And so obviously this Balanus trigonus was there first, and then the Balanus venustus settled on top of that. So we actually have a succession of settlement as it goes between ports. Okay, so this is Amphibalanus venustus. Here's some nice pictures of it. Okay, so how did we do this? If we look at this, there's some more story here, but here's a barnacle on top of a barnacle. Here's a barnacle that's on top of a barnacle that's on top of a barnacle. So you can, and then this is the paint from the hull of the ship. So we know that that barnacle underneath is the first one down. But also these barnacles overgrow each other. And so they can actually overgrow another barnacle. So if you find a great big barnacle with little barnacles under it, you know that those little barnacles were there first. Okay, so we can make these layers, and we ended up with something like 9 layers. This is Balanus trigonus again, with Amphibalanus venustus on top of it. Okay, and then we can measure the dimensions, the base dimensions of the barnacles to get their sizes. So I'm gonna sort of tip things ahead. This turns out to be Megabalanus cocopoma, and then these are Amphibalanus venustus. All the rest of these things on here, except for down here in the corner, is Amphibalanus amphitrite. That should not be a capital A. So we can then keep going through these layers and identify when things happen, when they got on. So it's a very, actually, it's an arduous process. And Ralph and I were doing this by hand under a microscope. He's my age. And after a day of this, we realized, okay, we're going to work 3 hours a day and we're going to quit or we won't be friends anymore. And it was, but in the end, we pulled them off. We dissected the barnacles and photographed them and published these figures of the barnacles. And we gave those, all of our material in the end, to the LA County Museum. But then when we did the analysis of those barnacles, so now we're going to talk about things that Grace Rowe, who's my graduate student here, and I do now, is that here are the base diameters of all the barnacles, okay, of Amphibalanus venustus. Okay, so then here, but there were dead ones and live ones, and they were in layers so that we could tell which layer died. And it turns out that all the bottom layers had died and that the new layers were on top of these dead layers. And so we could actually separate that out, and you can actually see there's a size frequency distribution based on the base diameter. So that's the frequency of the barnacles under this thing. And what you see, of course, is the bigger barnacles are more rare. But if you look at something like venustus and live, you can see these dagger-like, or trigonus, dead and live. You can see that there are these modes, these incredible modes. So the dashed line is the dead Balanus trigonus, and the solid line is the live trigonus. And so you see these, those are cohorts. Those are settling cohorts on the ship. And so they would go somewhere, the barnacles would settle, and that batch of barnacles would be there. And then there would be no settlement when they're in Woods Hole, because these are tropical barnacles. They can't settle on the ship when they're in Woods Hole. And the ship is mostly in Woods Hole. So it'd go to some place and then come back, and we could find the sediment layer. And we could count the layers that we had of live and dead barnacles. And then we could estimate their growth rate over time. But we didn't have a clock for that. We just said that they're different. We know that these larger barnacles are older than the younger barnacles, but there's no clock. We weren't there studying these barnacles over time. All we could say is there's this much difference between these barnacles. And then when we do but if we looked at the pattern of shipping of the trips of the Oceanus between ports, it was in Jacksonville, Florida on dry dock, then it went to Greece, then it went to Jacksonville, Florida again, then it went to the Bahamas and to the Port Everglades, and then it went to Gulfport, then it went to Barbados and Puerto Rico, then it went to St. Petersburg and Panama City, then it went to Barbados and Morehead City, then it went to Bermuda, and so on. So this is the latitudes of those ports of call. So the further down it goes, the lower the latitude, the more tropical the place was. And then here it went up to Maine. So that's, for the barnacles, that's not even as good as Woods Hole. But so that from the ship's log, we could put this clock together. Then we measured the base diameters and just said, okay, if we look at the base diameters and the cohorts of the base diameters, what impressed us was that if we lined them up with the timing of these things, that we got this very, very straight line. And so these are the live ones. So this is when it got to, this is February 26th, I think, whenever it got to here. 2 days before we sampled the ship, 2 days after it arrived. So this is when it's on the west coast here in Newport. And this is all the rest of this time is when the ship is in the Atlantic. Okay, and this, so if we just put this base diameters in the lines, it's this perfectly straight line. And then if we took the dead assemblages of the barnacles, this Balanus is not in the published paper, a story is coming. This also had a very, very straight line and matched up with these ports of call remarkably, so that there was an R square of 99% correlation. For such a small sample, what I was afraid of when we tried to publish this paper, and we just wanted to say, these are our results, this is what we found. And if this is true, then this is what we think. But the R squares, the correlations were so tight that I thought that it would send off alarm bells for any reasonable reviewer, including myself. If I'd received this paper, I would have really thought that was suspicious, but the linearity of it was just strikingly remarkable. So we did more things. In Woods Hole, we took the temperature of the water temperature of Woods Hole, which they record over time. There's a straight line when the temperature recorder went out, but there's these incredible seasons. And so the temperature goes down to 0 degrees, almost freezing in the wintertime. And it was some very bad winters. In fact, in the winter of 2009 was particularly bad and killed off all the barnacles, not all the barnacles, but most of the barnacles in the area. And it was enough that people published papers to remark about that. So then when we put this on here and we look at the temperatures, now this is a complicated graph to look at because this axis really represents 3 things. It represents the base diameter in millimeters of the barnacles, so small barnacles and big barnacles. It represents the temperature in degrees centigrade, so ranging between 20 degrees and 0, right? And then it also represents the degrees latitude. So, you know, they didn't get to the equator, which would be 0, but Woods Hole is at 41 degrees north. So every time the ship went somewhere, there's where it was at an excursion and another excursion and so on. And when we line these both up, strikingly high correlations, you know, there's almost no variation, right? It's too good to be true. So what this meant to us is that we could record where the ship was just by looking at the bottom. What is this? I went into some bad thing here. I'm not advancing. Let's see if I can do this. No, here we go. So this is what we found in Yaquina Bay. We found the layers. We're looking at it, Pacific to Atlantic, probable prior introductions to Yaquina Bay. This means that other boats have come to Yaquina Bay that had this barnacle on it. The Oceanus coming with these barnacles was nothing new. Nobody was watching. This is the first time anybody looked. So the reason that the barnacles were or were not here, the Oceanus probably had almost vanishingly low probability of having any effect on anything at all. It was just there, and we're able to trace something. But there is actually a lot of newspaper accounts of this. There was a lot of concern when this came out. Trigonus is a native species on this coast. Venustus is not. But when we look at the temperature range that trigonus can withstand, it occurs as far north as Monterey. And so if we look at its tolerance of 10 degrees, something like that, and we look at Yaquina Bay, the temperature is much lower than the northernmost location where Balanus trigonus is known to occur on this coast. So temperature is important in determining the distribution of these barnacles. And so totally logically, and maybe temperature is not the ultimate hammer that kills things, but everything associated with that is sufficient that we think to limit it, and it's a good coincidence with where the species occurs on this coast. So there was no harm in that, except that we could trace this. We had a very good measure, and same for Amphibalanus venustus. It can actually tolerate higher, Yaquina Bay has a lower temperature than the highest temperatures that it could tolerate. So probably the temperatures don't coincide. It doesn't mean it's not here, but easily could mean that it's not discovered yet, so nobody's looking. And so that we looked on the bottom of the Oceanus and found this barnacle has almost no meaning because it's not on a piling, it's not on a rock, it's just on the bottom of a ship that just got here. Okay, so then we wrote a paper, and the paper is port-by-port accumulations and dispersal of whole fouling invertebrates between the Mediterranean Sea and the Atlantic Ocean and the Pacific Ocean. That's all great. And Jim Carlton's, I think, maybe the most famous invertebrate zoologist in the world, in the marine invertebrate zoologist in the world. He's very, very widely recognized, and he was doing these barnacles and starting out on this. So after we published this paper, let's see, let's go on with this. In summary, the research vessel moved over 3 years. We could trace it. We could tell where that ship was, right? We could tell when it went south every time. And the age discriminator software we used was pretty crude for now, but it worked. And it was able we were able to analyze these data with that software and come up with these cohorts and make this analysis that would find these amazing things about where the ship went and every port of call. And survival continued even on these tropical barnacles when they went to a high latitude. They probably didn't reproduce, but they were able to survive there. So as a mechanism for introducing something, even if a ship goes to a very high latitude with things that are growing on it that can't survive there, if it goes somewhere else, it still can introduce them. And only the extreme oceanographic conditions killed it. So I didn't say enough about this, is that when the dead assemblage happened, it was preceded by the coldest temperatures observed in Woods Hole in many years. So it was a big barnacle die-off that probably killed them all. It made sense. Is a just so story. And then the other part of it is that we know already, but most people don't realize this, that every boat that moves between any harbor, between any place, carries something with it, that things are transported by everything we do. Okay. So that was a great story. Got the paper done, really proud of it and all that. Several months later, Jim wrote back to us and he said, well, there weren't just 2 species, there were 4. The what? So I know another biologist who gave a seminar here and talked about how his code, somebody asked him a lot of questions about his paper, and then they wrote him about how did he get his data, and then they want to get his code, and he said, this is really bad news. When you write a paper and that happens to you, that's bad. And so it turns out that he made some error in the code and clobbered his own data, and they got him, and okay, but it was all fixed. But this is science. We're supposed to be punching holes in each other. So, but here he's telling us that this totally wonderful paper has a ginormous hole in it, that there are 4 species and not just 2. And so here we go. Here's this wonderful story about these barnacles, and here it takes off to Greece. Well, one of the species that was in there is the volcano barnacle, Amphibalanus cocopoma, and it's a very large barnacle. And it's been introduced around the world from the Pacific Ocean. So no big deal here, but here's, I think this is a 2011 distribution of the species. And so species that are introduced are non-native. So that would be red. Species that are native are blue, that have lived there all along. And species that we can't tell if they're introduced or not are yellow. Well, it's not on here. We don't need to worry about that quite. But it has this patchy distribution around the world, cocopoma. But we said that the biggest barnacles settled on that ship in Greece after it was on dry dock. And Amphibalanus cocopomus is the biggest barnacle on the ship. So our conclusion was impossible. So that's pretty bad. Then on top of that is that the other reason why I want to give this talk is that Jim was going to come up here a month ago or a few weeks ago, and we were going to go get all the samples and go look at it and redo it. And then I was going to give this talk. But I got COVID and the samples were lost where we sent to the museum. And so it totally tanked. We couldn't do that. So all I know is that I had the 4 species. And so that's where we were. what does this mean and how bad are things? And so our thought was that the cohorts, whether they're a mixture of species or not, is actually even more interesting. And it's not just 2 species, that we're looking at 4 species and they're all behaving in this certain way. So our measurements of diameters were correct. It's just that we don't know what we were measuring then. We were wrong about that. And so the very largest barnacles, though, were this Megabalanus cocopona. So what I did was took all the old data back and reanalyzed it. And so what you do on this analysis is that the newer way we do this is that we assume, don't worry about what a standard deviation is, but it's a variation about the mean. So we always expect when we're measuring a cohort of people, of, I don't know, settling something, a fish or something, that there's a cohort, and that when they first start out, they have one kind of population structure. There's big and little ones and so on. And as they go through time, the relative differences between those aged populations changes in a coherent pattern. They die off in a certain way, so that sometimes the standard deviation can expand or to contract. But we expected a linear, we're demanding that whatever analysis has, it has this standard variation, pattern of variation about it. And then the other part of it is that we demanded that it has a coherent growth pattern. But in this case, because we know they're settling on ships at weird times, we can't have that. It's because we know that they don't settle at regular times. They settle when they go to the tropics or something. But we can take these analyses and look for the cohorts. And so the colored lines that are hard to see here are the theoretical lines, and the dashed black line are the actual values, the frequencies of the barnacles. And then what you can do is look for a correlation between those 2 lines. When you have a high correlation, that means that you've explained what the population looks like. And so it's not a factual thing. It's like parsimony. It's saying that we can explain what this population looks like. And so we find other ways to test it. And so this is where we're testing it with a clock that we think really exists. And if we're really wrong, when we go back to the Oceanus, the clock won't work. So that was the idea. And so Megabalanus cocopomus only occurs in Jacksonville, Florida, not in Greece. So it didn't get it there. It had to have gotten it in Jacksonville, Florida. We were off by 1 port. So the question is, if we moved it over 1 port, would it still fit? So we did that. And so what we did was we did these cohorts. We identified those, and they're hard to see here, but they're across here. And then these cohorts are lengths, different lengths of cohorts. And so it's hard for people to understand, or in general, it's hard to communicate this. The largest barnacles settled the longest time ago. So when you're looking at a graph like this, this is the earliest date on this graph, which is February 1st is when this was in 2009. Okay, and then it goes out here to December, which is, well, not December, but sometime in February in 2010. So this is closer to the present, and that's when you would find the smaller barnacles. The biggest barnacles were there first. They had the greatest length of time to grow. This is the death assemblage, though. This is not all of them out to 2012 when the Oceanus got here. We weren't asking about them. We didn't need to worry about those because they didn't have the really big barnacles. They didn't have the cocopomus on them, except tiny ones that we didn't have to worry about. So this meant that if we match up the clock, it seems to, uh-oh, here we go. Why did it do that? Okay, it skipped way ahead on me, sorry. Here we are. So then when we match up this clock this way, this missing port right there is, we shifted it back this way because of this distribution of cocopomus not occurring in Greece. Then we match up this and we get a very high correlation again. So even if we shifted it back, it's still matched exactly. So the story is that you want to ask questions. You don't want to believe in your own data. You don't want to, but we do that. If you're doing science, you do something, you get very excited about something, and then you tend to be an advocate of that idea. And that's very, very dangerous. That's almost always when you fail. In this case, though, We wanted to go back and fix it, and it turns out that this giant barnacle that was on the bottom of the Oceanus, it really messed us up. It made us think, made us reevaluate what we were doing. And even though I never set foot on the Oceanus, I never touched the hull, never was there, except that the divers pulled things off and gave them to me, and we analyzed these things. As that the ship, when I'm seeing it go a decade later, there's this poignancy of this story that happened, of this adventure that we went. Okay, so that's really all I had to say, is that you can look at these organisms that move around the world. They are very significant. The world is changing very fast, and it's kind of astonishing the ways that you can look and see how they moved, and even going forwards and backwards through time. So it's not just a matter of seeing them, you can actually witness them in other ways. And so I still think that our correlations are probably good, but we think people should challenge us. Okay, thank you. And questions. And I'm going to try to play this video. It did work. And if it overpowers us, we'll have to turn it down. But this is when the Oceanus came from Woods Hole to here, they made a time-lapse video of it going through the Panama Canal. And at first I was thinking, oh man, they didn't do a video of the whole trip. Well, when you're on a great big ship going across the ocean, how many times do you want to see the big blue sea out there? How many frames of that do you want to see? You got to have something happening. So they did it just through the Panama Canal. So let's see if this works. This is a ship that is no more. It's not going to be used for anything. It'll be scrapped or something probably. This is Lake Atun. It's fresh water. All the organisms on the bottom of the Oceanus were marine organisms. They survived going through the Panama Canal. Lake Atun is full of introduced species from all over the world. It's still there. You can still see it. Thank you.
All right. Thank you so much, John. I want to open it up to questions, and I'm actually going to look over at Roseanne. Do we have questions online at this time? None at this time. Do we have questions in the room? Yes, we do. Okay, we'll work through some of these.
That was great, John. Really, really interesting. So you discovered these extra species that you didn't know were there before. So would these species have been overlooked in some of these harbors?
Of course.
I mean,
The-
so there's more confusion going into that. And the other question I had is, have there been temperature tolerance studies done on all these species?
Have there what?
Temperature tolerance, because they go through, that seems to kill them off. Are some more able to withstand such a long distance temperature change?
Well, temperature is something, in biology that's used so often because we can measure it, right? If pH was easy to measure, we might look at the world with pH or something. But temperature does, it's definitely associated with the distributions of a lot of organisms. So it's a very valuable kind of a measure to look at where species are. And it does appear that when the temperatures were very cold, close to zero, these tropical species did die. So maybe the temperature did kill them, but all kinds of things happen when the temperature gets close to zero. But the other part of your question,
ranges.
of the other species could still be here that we didn't look, but we've looked for them. And we actually, what happened is when we looked for barnacles here in Yaquina Bay, we found other introduced species that weren't reported here before. So I didn't want to even talk about that. And so there are very few people that can identify barnacles. So it's no surprise that when somebody that can identify barnacles goes someplace, they see things that nobody else can see. And so there are very few people that do that. There are very few people that know the algae of the coast, right? So they don't see something that's remarkable that shows up. You have to find it backhanded. And so most of the introduced species that I've ever written about or known about, I found out about them from somebody that was not a zoologist, that told me about something that had a remarkable finding or something. I didn't find it, even though I go look for things. And when I go look, I find things there, but I can't be everywhere. And so the problem is, I don't know if it's a problem even, is that the world is homogenized and we don't even know how much. And it's because of these things. The Oceanus had nothing to do with that. It's just that the Oceanus was just another ship that was going from one place to another. And that's the reason why we wanted to look at it.
All right, we have a question online.
Okay, the questions are, this is a double question. Does the length of time the ship is in port affect chances of barnacle recruitment? And the second part, how long does exposure needed for a barnacle larva to settle on the ship's hull?
Those are really good questions. The barnacles are, they settle within minutes. They're really thick in zooplankton in a lot of these ports. But they're in the wintertime, they're not there. They don't settle, so that if you go to some ports in the wintertime, you probably wouldn't get those barnacles. And then the Oceanus, when it was here, only 2 days, we found species from Yaquina Bay that had settled on the Oceanus. So there was an isopod that was swarming in the bay, and it settled all over the bottom of the Oceanus. And we know exactly it didn't come from the Atlantic Ocean. No, this is our. And so we knew that, and because we knew enough to know that. But since even then, there's another isopod that showed up in the bay that that we never knew before, and so that was confused with that isopod even. And so these, it takes no time for some things to settle on the bottom of the ship. And I only talked about barnacles, but there were probably about 10 other species that were on the ship that we found. And this is an extremely carefully maintained ship, the best, the highest quality maintenance of a ship. And yet we could still find those things. Even if they were in a port for a couple of days.
Okay, one other question here. Do barnacles have seasonal settlement?
Yes, they do. So I can't speak for all of them, but if you're in the tropics, the barnacles pretty much settle all the time. But the higher the latitude, the more seasonal the settlement of the barnacles tends to be, because there are cold and hot times. And, runoff and rain, various, all kinds of reasons for that. But yeah.
All right, I think we have some questions in the room.
Yes, I wanted to ask of the various species of barnacles which have been introduced around the world, which of these do you think is having the greatest impact on the regions to which it has been introduced?
That's an excellent question. Well, maybe they all have some impact, but it's not really measured directly. It's not like somebody has a number and says this is the GDP of barnacles, but the amount of money that is spent for the anti-fouling paint and for hauling ships up and scraping them off, and the substratum that they scrape off the bottom of a boat, any kind of boat that's been in the water for a long time, the major substrate in marine systems are barnacles. And so the expense of taking care of those barnacles is truly enormous. There were always barnacles there were barnacles on this coast. There just wasn't the great diversity of barnacles that we have now. And so probably I'm just making this up, I don't know the answer, but very likely the amount of time and energy or resources that are needed to deal with barnacles now is greater than it was 500 years ago. So Columbus sailed across the ocean blue in 1492, and he brought 500 species of colonists with him probably. And then he came back to the old world, the whole lot of other things that were drilling holes in his ships and everything else. And he even had to abandon one of the ships because it was in such bad shape. So, and Columbus wasn't the first to go across the ocean or sail around. They were doing it way before that. So yeah, the barnacles are not something that are, I don't know, as impressive as something that like a shipworm that drills a hole in the wood, but they definitely slow ships down, and they're very costly for that. Yeah.
Roseanne, any questions? Okay, I think we have another question up here that I did a flyby on.
Is there any interest in redoing this study with the Tawny as it goes from dry dock in Louisiana, right? Like from the Gulf back through the Panama Canal to Newport, or as it's going through its paces?
Oh, go in the reverse?
Yeah.
Oh, well, that would be fun. But somebody just, no, it's really fun. It was really, even though Ralph and I we finally agreed to get along. It was so intense to do that layering. It's like, we're really good friends, but it's like, it was sort of falling apart. And so I'd measure something and he wouldn't record it right. And it's, you did what? I've got to start again. So, but it's, to actually do the layers and to try to figure out where it was like this incredibly wonderful puzzle to undo. And I think we did. But so the Oceanus was gone, and the sister ship to it was the Wacomas was right there. And we were going to compare them. And the Wacoma was really uninteresting. It's just covered with barnacles, a couple of inches thick. It was mountains of barnacles. And we knew what they all were. And it's like, we don't care. And so we just stopped, didn't do it. But the new ship, the Tawny, that's coming, is going to come from the Atlantic Ocean to here again. So you have a model. So you could do it again. Maybe, I don't know, if you could get permission to sample it again. It would be interesting to do that, to see how they move. And it's not like we're going to, I don't know, you're not going to get the Nobel Prize for that. But what we want to know is how these things move and how fine that is. And what we think from our study is that we were able to see it's extremely fine, that there's always something that's moving. And we kind of thought that, but we're even then kind of amazed that, yeah, it really is like that.
Okay, I'm going to be selfish and ask my own question, John. So can you talk about possible differences in technology in the way that we deal with fouling between the Oceanus and the Tawny?
You're going to have to repeat that. I'm sorry.
Sure. How much has changed in the way that we deal with ships moving across the world between the time of the Oceanus and the time of the Tawny? Is there changes in the paint? Is there changes in the process to?
That's a really good question. Well, the Oceanus was extremely well maintained, right? did all the right things. They had the best fouling paint. They had it on dry dock only 2 years before it got here, and they had a regular maintenance schedule. So it's almost like saying that's as clean as you can get right now. So there's ballast water exchange rules in place already, and I don't know, they might make better paint, they might make better, faster ships, but it's like that at that level of, it's like that's as good as you can get, and they're not at fault. It's just the very best we could see things still moving. The other side of that, though, is that ships are maintained for economics, among other things, but there's a much more consciousness of cleaning up ships. They don't like to have dirty ships moving somewhere. They took the USS Missouri, towed it over to Hawaii, but they parked it in the Columbia River for a month or two before they went there, only because they were worried about introduced species. So those changes are happening, and it does, and probably very likely to make a difference that they do that. And so I guess my feeling is that I study introduced species. That's what I do. And I'm not an advocate for introduced species, but what I'm trying to understand is what do we do about them? And the only way you could do that is to understand what happens and to understand what they do. And so the other parts of it are way beyond things that I could imagine that other people could do. But in your case, what you're saying is that the ships, what you're mentioning, is that they really are making very big efforts to stop moving things around the world. They're still moving, but it's the right thing to do, but there's still a lot more to do.
Thank you, John. I think we have another question.
You mentioned a replica ship with a Golden Hind, was it, that went down the West Coast? Were they in the Columbia River in freshwater long enough to kill the marine species? Or does that tidal exchange give them saltwater every day?
That's a pejorative question. Of course it was. Well, no, we assumed it was, and they assumed too. I think they thought that they parked it way up in the freshwater but no, your question is, but we didn't ask a whole lot of those nasty questions because we wanted them to cooperate with us and let us bolt things onto the ship and things. But that's true, is that the USS Missouri that I just mentioned, they took it to Hawaii, and they found a whole lot of species that were still alive that had settled on it in the Columbia River that made it to Hawaii. And so nobody knows that any of those survived, but it's like that's a big ship. It would have taken a fortune to clean that stuff off. So they were doing this quick thing. And even in Yaquina Bay, they were going to have a ship breaking here in Yaquina Bay 15 years ago, something like that. And they were going to tow ships from Mare Island Naval Shipyard and from Suisun Bay and San Francisco Bay up to here and break them up. And one of the big concerns was that they would bring all those things that are introduced into San Francisco Bay into Yaquina Bay. So what they did was they came up with this incredible, ingenious thing. Why not break up the ships that are there that were built at the Mare Island Shipyard at the Mare Island Shipyard, where they needed to have jobs and where they had a dry dock and everything already built. And so complaining was actually quite good for us and for them.
All right. Any other questions online? All right. We got another question in the room. It'll take me a second to get there. So hang on, everybody. Hang on just a second. You need the mic.
Barnacles in the Atlantic coast, or are they just ships over the place?
Yeah,
It just seems like there's such a hodgepodge now that it's like, it just seems like if the Atlantic barnacles come over to the Pacific side, then they're going to cause a big disruption on our side. Other species will be.
Well, that's a good question, and it's a really common question. Why hasn't everything already moved around? Why isn't everything already homogenized with the graph that I showed in the very beginning? How come everything isn't already everywhere? Well, the other part of it is we're changing the world. And so these things are being bombarded onto these changing environments. And for 100 years or 200 years or 300 years, they can't make it. They can't survive there. And then eventually there's enough change that they can. And so the world's still quite diverse. In fact, the planet probably has as great a diversity as it ever had in the history of the planet because the way the continents are done. But humans are doing a good job of homogenizing it and making it less diverse.
All right. Oh, one more question in the room here. Hang on, John.
Have there been any studies looking to see how maybe biological agents like barnacles grow in whales or like other large organisms, like super transient species that move around maybe into other oceans or around those areas? Have studies been conducted to see if they also have the same like transmission rate of barnacles to new areas?
I guess you spoke clearly, but I didn't hear it well. But I think you were saying, are there other species like barnacles that are getting moved around like them and associated with them?
I think we're talking about barnacles on something that's biological itself, like a whale.
On whales and things like that? Yeah, of course. There are barnacles that get on whales that also, I don't know, and this is like, I don't know. So I'm going to start that. But why couldn't whales have barnacles that get introduced because of whales? And so that does probably, but whales move around and we change the distribution of whales, we kill them off, they go and expand and things like that. So it's possible. But most of the things that we've seen on whales and turtles are have always been there. But now we have plastic debris that's going across the ocean that's made these garbage bridges for things to drift across the ocean on. So something like a turtle or a whale that's associated with that is exposed to something that it never was before. And so there is this, we're just discovering what all this is. And so we thought that things couldn't naturally drift across the ocean until about 2012, when the tsunami debris hit us and we realized that there's this garbage bridge that's carrying these things. We didn't know. We should have known, but we didn't know. We weren't even looking at it. We thought it was totally trivial, that had nothing to do with introducing species. But now we see that there's a garbage bridge that goes across the ocean like old ships and things like that. And the whales that are out in the ocean that are exposed to those, sure, they can pick that stuff up.
Interesting questions, everyone. One more check online. Anything, Roseanne? All right. Oh, we got one more question here. And this will be our last one.
Yeah, I guess I had a couple questions that might lead to one question. I was curious if barnacles are Euryhaline, and I also was curious if they're selective eaters, if that's really understood, if they're selective eaters, and if they have like a specific diet, like something specific they like to eat, and how that might play into where they're found and how they're able to survive as opposed to temperatures or other factors.
I think your question is there selection on the barnacles? On what?
Are they selective eaters?
Diet.
Yeah.
Oh, yeah. So why not? Sure. Barnacles are different. They must have different diets. How come they're all just out there in the soup, right? So probably nobody's even looking, right? Is that why wouldn't barnacles establish somewhere because the right kind of phytoplankton is suddenly there, right? Or it disappeared, whatever, something like that. So the world is changing and we're not able to measure and see all of this. But of course, that makes total sense. Why not? Yeah, nobody's looking. Good idea. Good question.
All right. Can we give John just another round of applause, please? And it sounded like about 17 different possible research projects was presented here today. So just keep thinking about those. All right. Thank you so much, John. For everybody online, thank you for joining us. We'll see you next week.
Good job, John.
Thank you.
So my question only, I didn't get it out. I mean, the first part you answered was great. The second part is what is the widest ranging barnacle? And it must have a huge temperature tolerance if it's so wide.
Well, it depends on how long it is. A tropical barnacle. Oh, yeah, a tropical barnacle. Turn it off. A tropical
All right, for everybody online, we are going to end the presentation. Thank you so much.